Hey everybody, today we're going to be talking about mitosis and the process of how a cell has to actually divide. We ended last time with why a cell needs to divide before it gets too big, but we didn't really focus on what that process is of how it has to actually go through the division. So the first thing that we focus on is the cell needing to double everything. More importantly, we're going to be focusing on our chromosomes. Now, a eukaryotic cell is a cell that has a nucleus. We talked about that first semester. Now, the chromosomes are our genetic information. So all living organisms must have a complete set of genetic information. That genetic information is our DNA. And like we said last semester, DNA contains all the instructions of how to run the cell. It tells my kidneys how to function. It tells my skin how to repair itself. It tells my body what I need to look like and how I'm going to function. Now, DNA is very hard to see. So what DNA does is it packages itself, so it squishes itself down, and it forms these chromosomes, this X shape. Now, we cannot see DNA in this double helix form, but when it condenses down into a chromosome, we can see it. And it makes things a lot easier to separate. So think about if I had a ball of yarn and I untangled it all and I had string everywhere. It's going to be really hard to separate that DNA because I have this big stringy mess. But if I take that yarn and I ball it up and then I just cut that ball in half, well, it's going to be easier for me to now have two separate things. So it's making it easier to split during cell division. So we're going to focus on this eukaryotic chromosome, all right, this DNA. So DNA is found in the cell nucleus. The nucleus protects it. In the human body, we have 46 chromosomes. Now, chromosomes or DNA can be found in multiple forms. The first form is going to be chromatin. Chromatin is uncoiled DNA. So my DNA is still tightly wound, but it kind of is still relaxed. We're not forming that X yet. I say that this looks like a bowl of noodles or a bowl of spaghetti over here. Now, our DNA can be found in this form during interphase. Interphase is going to be one of those first phases of mitosis, of cell division. And with interphase, the DNA is still relaxed here. It's making a copy of itself, but it's still being very relaxed. The next form is going to be a chromosome. This is coiled DNA. And this is how DNA looks after division. So after it starts to divide. The replicated chromosome is what we see here. So we go from spaghetti to a chromosome to a replicated chromosome. Okay, so this is our coiled DNA, and this is DNA after the S phase in interphase, which we're going to get into a little bit more. Okay. So we have three different kinds. Now, if you look at this picture over here, we have what's called a sister chromatid, and that means that these guys are sisters, that they are identical. Okay. So when we talk about the eukaryotic cell cycle, the cell cycle itself is a whole series of events that allows our cells to grow and divide. We said in the previous lecture that our cells do need to grow a little bit in order to accommodate for doubling everything. But then the big part is that it's going to divide. Now, this is going to happen in somatic cells. These are known as our body cells. So every single cell in our body except for egg cells and sperm cells. So those are these sex cells, which we're going to talk about in meiosis. So the only things that are going through mitosis are my body cells, my heart, my skin, my liver, my muscles. All of those things are going to be going through mitosis because they're going to make exact copies of themselves. Now, in the cell cycle, we have two main stages. We have interphase, so my cell is not dividing during this phase. Interphase is made up of three phases, G1, S phase, and G2. The M phase is going to be where my cell is actually dividing. This has four phases, prophase, metaphase, anaphase, and telophase. A way to remember the order of all of this is I passed my algebra test. I for interphase, past for prophase, M for metaphase, A for anaphase, T for telophase. All right, so on this, we're going to have a few things going on. So right now, I don't want you to copy anything just yet. So your circle, I have a few things written, but you guys are going to be editing your circle a little bit differently. So the very first thing I want you guys to do is on that outer ring, I want you guys to write interphase. And then on the bottom, we're going to write cell division. Okay, if you guys want, you can copy, you can use color pencils and do what I have and have it kind of color-coded like this. So in interphase, we have our three phases. We have G1, we have our S phase, and we have our G2 phase. In cell division, we have mitosis and cytokinesis. So if you look at interphase takes the longest. This is the non-dividing stage. This is where my cell is going to be getting ready to divide. The cell division is much shorter, so it goes very quickly. Now in interphase, we have our first one, G1. The cell is going to increase in size and it's going to make organelles for itself. So it's going to start to increase and say, all right, do I have enough ribosomes? Do I have enough mitochondria? And if it doesn't, it's going to make sure that it's ready to be going through cell division. The second phase of interphase is the most important. This is S phase. This is DNA replication. This is going to take a while because it is so important. That's why interphase is such a big part, because DNA replication needs to be done correctly. The last one is G2. G2 is the cell is getting ready to divide. So it's going to grow a little bit more and it's going to make organelles for the new cell. After that's all completed, we're going to be moving into cell division, and that's going to be mitosis. So the first stage of mitosis is prophase. We're going to put a P. The second is metaphase. The third is anaphase. And the fourth is telophase. After telophase, we have then cytokinesis. So if I were you, I would start here, and I would draw an arrow going this way to show that we start at G1 and we are going around clockwise. Okay, we're going to go through all of the cell division and mitosis sections in just a few minutes. But please remember, interphase is extremely important. It is the longest phase because all of these things, increasing sizes, making organelles, copying DNA, this all is a very long process. Here's another picture just to kind of show you. We have G1, S, and G2. These are interphase, again, very long. We then have our mitosis and cytokinesis. This is the cell division, prophase, metaphase, anaphase, telophase. So with interphase, this is 90% of the cell cycle. 90%. It takes the longest amount of time. We said there's those three phases. G1, the cell's doing most of their growing. The S phase, our chromosomes are replicating or copying. And then G2, we are preparing for M phase. That's that mitosis phase. So we want to make sure that we have organelles that are required for cell division, that those are made and ready to be going. M phase, like we said, M phase consists of two stages. Mitosis, this is where we're dividing that nucleus in half and getting rid of, separating that DNA. That includes prophase, metaphase, anaphase, and telophase. And then cytokinesis is that we're actually splitting that cell in half. We're cutting the cytoplasm. So cytokinesis is we're dividing the cytoplasm. The video below is just kind of showing an animal cell going through mitosis. You guys can click back and watch that if you would like. So the stages of mitosis. The first is prophase. Now, in cell division, we said before, cell division happens very quickly, but these phases do range in time. So prophase, we are preparing. Prophase, prepare. It's the first and the longest phase. So in this, our chromatin thickens into chromosomes. So here is our nucleus. Before, our DNA was in that spaghetti chromatin-like stage. Now it has thickened up and is now in our chromosome form. Our chromosomes are these little X's here. Now, if we look, the second part of prophase, we're preparing, we have to get these chromosomes out because we need to separate them. So the nuclear membrane disappears. On the side here, if you notice, we got these little strings and these little tic-tac looking like things. Excuse me. These are our centrioles. The tic-tacs are these centrioles. They are going to start making spindle fibers. That's what these strings are. So we have our centrioles and our spindle fibers. What they are going to do is they're going to help pull everything apart. So what they do is they start to get ready. They are preparing. So my centrioles are going to start to move to the opposite ends, and the spindle fibers are going to get longer. So we have two main things happening. We have our chromosomes are all set, ready to go, and our nuclear membrane is disappearing, and our spindle fibers are getting ready to help. The second phase is really, really short. This is metaphase. They are lined up in the middle metaphase. If we notice, our chromosomes are now lined up all along the middle of the cell. Now, I have sister chromatids. Again, our chromosome, our X's, we call them sister chromatids because sisters, if they're twin sisters, they are identical. They want to be next to one another. So they line up in the middle of the cell, and my spindle fibers attach to either side of them. The next phase is anaphase. We're pulling away or apart. Away for anaphase, apart for anaphase. So my spindle fibers, they've grabbed hold of the sisters or my chromosomes, and they rip my sisters apart. Poor sisters. So the sister chromatids split apart, and they're getting pulled to opposite ends. So those spindle fibers are kind of like reeling them in. The last phase is telophase. I like to say telophase as da, we're done, or two for telophase. So what happens is after anaphase, they pull them to opposite sides. My spindle fibers start to fall apart because we don't need them anymore. My nuclear membrane starts to reappear because we want to protect these chromosomes. The last is that they're going to start to loosen back up. My DNA is like, okay, I can relax. I can go back into my relaxed form, which is chromatin. Now, if you notice, I got these dotted lines. It's because now telophase is going to start to pinch in the middle to make two new cells. And that's what happens with cytokinesis. So in telophase, we start to have this pinching in the center. And then once it pinches, we get two new cells. So the cytoplasm is dividing in half. It's usually happening at the same time as telophase. So as telophase, the nuclear membrane's reappearing, the spindle fibers are going away, the cell is pinching in the middle, and boom, cytokinesis happens very quickly. Now in animals, the cell membrane's going to pull itself in the middle and kind of create that peanut shape until the cell is pinched in half. In plants have a cell wall. They can't pinch a cell wall in half. So they're actually going to form a cell plate. Essentially, they're building a new cell wall. So again, here we have prophase. Prophase, it's preparing. My chromatin has thickened up into my chromosomes. My spindle fibers are appearing. My centrioles are pulling those spindle fibers off to opposite ends. And the nuclear membrane is starting to disappear. Metaphase, they lined up in the middle. My spindle fibers are starting to attach. Anaphase, they're being pulled away from each other. The sisters are being pulled away. They're saying, no, sister, come back. And then telophase, we're starting to pinch in the middle. Spindle fibers are disappearing, and my nuclear membrane has returned. Well, this is what's happening. Animal cells, they're pulling apart. And then plant cells, they're building another wall. So our body is constantly going through this process. Why? What is the point of mitosis? Well, mitosis plays a huge, huge role in our bodies. After you have an injury, we need to make new cells. So when you fall and you scrape your knee and you have a scab, your body is trying to make new skin cells. So your body is making identical copies because we want our skin to look the same in that area. We want the same color, the same texture, everything. We need to make new cells when that happens, because if we didn't, we would have a bunch of open wounds on our body, and that'd be super gross. So we need mitosis in order for that to happen. When you're growing, think about you as a baby versus you now. you're a lot taller, you're a lot bigger. Our body needs to make new cells as our organs get larger, as our skin starts to stretch out. But if your skin didn't make new skin cells, your skin would be pretty thin and probably be ripping very easily. So we want to make sure that our body is going through mitosis so that we have new skin cells and we have more cells so that our organs get larger. And the last one, replacing old blood cells. We need blood in our body for our body to function and work. And our blood cells do eventually die. So we need to be able to replace old blood cells. And again, we are making exact copies of ourselves. So we started off with one cell and we made an exact copy of it. So now we have two cells. So like I said before, our bodies have 46 chromosomes in each cell. So when my body makes a new copy, that new cell is identical. Same type of DNA, same amount of chromosomes, 46. So if you have any questions, make sure you guys are asking. You can also watch the mitosis videos that I have posted on PowerSchool if you're still having a little bit of trouble understanding.